Please. First of all, you're all very welcome to this research seminar given by Dr. Jonathan McCarthy. And Johnny is a graduate of the School of Psychology here, and he went on to do his PhD in King's College in London. Then he went on to do postdoctoral work in Brown University in the US, and then he returned for more postdoctoral work, which will culminate in a lectureship in King's College. So it's a glutton for punishment, and you're very welcome back to UCD.
Thank you very much for the invitation. I should probably tell you something now, in that most of my background currently has been in image analysis and statistics. So if I go off on one that uses too many acronyms or anything like that, by all means shout, because it is a tendency. But some of the things you're able to do with this. So over here on these images, you can see two templates. So these are kind of average kids, so we're not giving away anyone's identity. But this over here is the template image of a 1.5-year-old. So this is a median of about 50 1.5-year-olds. Over here is template of a 3-year-old. So what an indicative 3-year-old would look like. And the important thing just to take from this is that you can image them. That's fine, but you can also tell they're different just by looking at them. So does imaging really add anything to any of these things? So it's a very expensive way to develop behaviour. So if you're trying to figure out why you'd even bother imaging the developing brain, and quite a lot of the stuff we know about how the brain develops is from post-mortem, and actually that's the only way we can really tell, or from animal work, you're trying to start looking at how typical individual variation happens. So you're trying to see why one child might be developing at a faster rate than another child, and you're taking from it a completely biological point of view when we're doing this. So quite a lot of what we will be talking about is assuming that quite a lot of behaviours are underpinned by biology, but actually most of the biology we're looking at is genetically programmed, so it's going to happen irrespective of the environment. So the differences we see in inter-individual variation in biology are tiny, and whereas most of the individual variation you see in behaviour is going to be environmental. So there are certain things that we do know that makes looking at early development important. So I work mostly in a preterm group, and about 1 in 15 children are going to be born preterm. And of those children, about 15% will have very subtle behavioural disorders, so things like inattentive subtype of ADHD. They've double the risk of autism. That's not huge, that's still only 3%. There's about double the risk of epilepsy, and this is an environmental insult, really. So the child is born preterm, and it's their environment changes. A lot of neurodevelopmental disorders are occurring probably at this stage, so you might be able to diagnose autism at 3 or 4, but you can kind of assume that something's already happened at an earlier stage. And there are quite a lot of very subtle perinatal injuries. So if you look at something like epilepsy, quite a lot of the abnormalities that lead to the seizures, even in adulthood, are a function of something that happened when you were very, very young. If you look at this map, so over here is a heat map of the US, and it's from the Centre for Disease Control, which presumably still runs. So if you see here, at the top, it's the proportion of children in any given state who are diagnosed with ADHD. And the thing to take from this isn't that, different states have different incidences. It's the fact that different states are probably diagnosing at different rates. So some of that is probably the actual incidence of ADHD is probably the same across all these states, but there's different things that are happening in the society that's altering how it's diagnosed. So it's not all behavioural. The important thing for me to take out of this is the proportion of all children medicated. So at any given time, up to 9% of children are medicated in states for ADHD, and that is going to influence brain development. So even it's being reflected in increasingly big studies. So we were talking a little while back about how a lot of studies are becoming multi-site. So to collect an MRI costs about £500. And to do this at a single site, so if you're looking at 1,000 kids, you're looking towards £500,000 on a grant, and that's not including staff or maintenance or writing up or anything like that. Some of the newer studies are moving towards 20,000. So there's the ABCD study in the US, which is 20,000 children being scanned across lots of different sites. What we're doing in Kings is the E-prime study, which is 500 children who were born premature, and we're following them up longitudinally. The Developing Human Connectome project is imaging children fetally and imaging children at term just after they've been born. So this is quite large studies. The study I'm going to talk about today is the Brown study of infant brain development, which is a single-site study where we just collected lots and lots of kids over a short period of time. So when looking for biological bases for these diseases, which is what everyone's trying to do, whether they exist or not, there are quite a lot of indications that there's a biological basis. So if you look at head circumference, just something as simple as a head circumference, like wrapping around a tape measure, you'll see that at an early stage, children with autism tend to have a larger head circumference. If you look at this plot over here, you probably think to yourself it's nonsense because the plot has been fit quite badly at the beginning of the age period, but it's been reproduced. So this is a very highly cited paper. But the plot showing this growth curve going down at the beginning could easily be created just from the children at the early age, but actually it is something that reproduces. And the reason why you might be interested in the fact that there's brain overgrowth at an early stage is most of the brain growth that occurs is happening in the first 2 years. So between the ages of 0 and 2, your head goes from about 20% of adult size to about 80% of adult size. So that's like, and overwhelmingly that's happening in the first year. And there are two processes neuronally that are driving this. There's lots of other stuff, but if you're just focusing on neurons, there are two processes that drive this. One is synaptic development, so synaptogenesis, so this is the connections between different neurons and how they lead to communication. So they call this synaptic exuberance, I think, depending on if you're an old school anatomist, where you have huge amounts of synapses between lots and lots of different neurons, and that's too much. It's too much communication and it's not efficient. So as you use certain synapses and certain connections more and more, they become reinforced, and the ones that aren't being used start being pruned back. And that's also happening in overlapping periods in the first couple of years of life. The other big process is myelination. And this is also heavily related to how the brain is connected. It's not related necessarily to neurons. Most of your neurons at birth are the neurons you have now. Nothing's really going to change about that. So myelination is if you have this wire here, it's insulated, and without that insulation, the conduction velocity is less. So your brain starts to myelinate in a really programmed fashion, and that leads to much more efficient circuit talk. So just the way different regions of the brain are chatting to each other. The interesting thing about myelination is it continues throughout life. So if you think of someone who has multiple sclerosis, sometimes they have lesions, and that specifically tends to be attacking myelin, and they recover from that. So hopefully they recover from that. So those regions remyelinate. The myelination also changes in response to how different regions of the brain are communicating. So if you think of EEG, you get different frequency responses to different activity, and myelin will occur depending on the frequency response. That said, the degree of myelination that occurs according to that frequency response is really subtle. So if we look at this image here, if you're doing imaging, this is kind of depressing. So this is a histology stain from about 108 years ago, that shows really not much more than I'll be showing you today. In that, if you stain, so if you look at a 3-month-old, the only bit that's coming up dark here, this is a stain for myelin, is the internal capsule. So that's the bit that goes from the thalamus to motor cortex, and it helps with motor movement. So at an early stage, that's myelinated. That's actually myelinated usually at birth. As you move older and older, you see more of the brain starts to get dark. And that darkness is indicating that more and more myelin is being produced, and that just leads to more and more efficiency in the circuits underlying. So if you think of the difference between a child just after they've been born and a child at 2 years old, it's a completely different game. And actually, you can map out the cortex this way. So you can map out the cortex based on when the area is myelinated. So places like primary sensory, so auditory cortex, visual cortex, and motor cortex are the first ones to myelinate. Then you start seeing kind of sensory association cortex, so You might see the visual word form area, or any of these ventral temporal areas are next, and then eventually areas that you'd associate with executive function. So these would be the last. So if you think up here, spatial working memory, if you're doing a spatial working memory task, they're the areas that activate, and they're the last to myelinate. And these are skills that you continue to build throughout childhood and even in early life. This is roughly the same as what I just said, but the period from birth to 6 years is quite dramatic. So it's biologically dramatic, it's behaviourally dramatic. It overlaps with when we're expecting to see periods of, the onset of neurodevelopmental disorders. And most of what I do is atheoretical, so we're just looking at stuff. So it's observational and it's very exploratory. But there are certain things that we would be expecting. So there's pretty solid evidence that the interaction between brain and body and brain and behaviour probably changes as a function of development. Some of this literally has to be the case, but so that's probably the only theory. So this is Mark Johnson's interactive specialization idea, which is that, for instance, as you begin to develop words, there's a specific area in the brain in adults that you associate with the visual word form area, so it'll activate when you see a letter. And that's there when you're born, but it's not used for words. We only really systematically use words for the last 200 years or so. So it just gets co-opted to use the function that you later do. And that's the kind of interactive specialization we're talking about. A similar area on the other side would cover face perception, for instance, and they're all kind of symbolic processing areas. So as I say, I'm going to focus on the Brown study of infant brain development. So this was the study we were working on. It's kids aged between 0 and 8 years, and it's called an accelerated longitudinal design. So this is probably slightly new for grants. So an accelerated longitudinal design is you start scanning children throughout the lifespan, but you also follow them up. So it's a longitudinal design, but accelerated because you can look at the entire period through childhood. So in our case, we're looking at about 500 kids. I think it's up to, well, they call it new. So it's come up quite a lot in.
Maltese developed that design, I would say, 25 years ago.
I know, so they've started, that's why I say they call it new, they've started using it for imaging studies all over the place because they're trying to reduce the cost instead of doing fixed.
He did lifespan longitudinal designs with, it's a cross-sectional interspersed with a longitudinal, so that you end up with a huge amount of data. It's a very good design.
Can be quite noisy is the downside of this, but so it's come in as, so I say they call it new. It's kind of come in as a kind of a buzz term in quite a lot of.
He called it cohort developmental, I think, or cohort sequential is what he called it.
I think you'll probably find, even with the imaging, most of the stuff I'm saying here is probably a rewording of what it originally was said. So in this case, we are purely focusing on white matter development. So we're looking at myelin. And when I'm talking about cognitive assessment, what we do is going to be very, very coarse compared to properly developed studies, because we usually have probably about half an hour to an hour with any child. So we do behavioural studies using a developmental battery that's been normed in the US, but it is quite a blunt instrument. In our case, we're using the Mullen scale. So initially, we're trying to do two things. So we're just trying to replicate. So
yeah, I was just wondering if it's just structured MRI, were they under anaesthetic, the children?
No, I'll explain how we did that. Yeah. No, they're all asleep. I lost a lot of sleep. I didn't have grey hair before I went there. So we scanned only at night. I'll show you roughly what we're doing. But for instance, in St. Thomas's, where I'm currently working, the infants, they're usually neonates, will get a scan usually for clinical purposes, so they have a mild sedative at the same time. But if they're infants, they don't really need to be sedated that much. You rarely, for a research study, we wouldn't really have the opportunity to sedate, and we wouldn't really want to, just because the risk is unnecessary. Well, probably not in Europe, but in the US you might be able to get it through. But So how do you scan in this age range is probably the next bit. So I'm going to be a little broad for the moment because it entirely depends on what you're doing. So when you're looking at, so if you want to do a task or something like that, you obviously can't do it with an asleep child, but actually you can probably get away with it in some ways. So the degree to which you can scan depends on the age, it depends on the type of scanning you're doing. We're using MRI. And structural MRI is actually alright because structural MRI, as a sound in the background, is low frequency. It just goes. But we left to the very end functional MRI, which is just. So if the child's likely to wake up, they're going to wake up during that. So that the same applies for diffusion MRI. As a general gist of experience from various different people, I've color coded it according to what's straightforward and what's not. So structural MRI actually is probably pretty straightforward throughout the lifespan. MEG is difficult enough with infants because the contraption itself is fixed. EEG is actually, I'm being a bit harsh, with a toddler, putting on an EEG net is either it works or it doesn't, and that's it. So I call it red just because if there's a child who doesn't want to do it, there's no way it's going to happen. Functional MRI is very loud, so if you want to collect data asleep, it's probably okay. But either way, the thing to keep in mind is that all of these kind of imaging acquisitions are just significantly more difficult in children than they would be in adults. So if you look at like the scanner over here, it's objectively an odd environment. You can't, kids aren't exactly going to get a kick out of going into it. So what we usually did was scan at night. So we tried doing daytime scans a little bit, but it didn't really work because you're hoping to match onto nap times and things like this, but it didn't necessarily work. Another thing is preparing the child was almost not nearly as important. It's just making sure the parents were comfortable with it because they tend to be, as you would be, quite anxious if you're putting your child into that. So people are quite interested, in brain development generally, but you just had to make sure that the parents were comfortable, because if they weren't comfortable, even if they said they were, there was no point in doing the scan. What we had was right beside the scanner, so if you imagine that door is where the scanner is, we had pack and play in the back if there were young kids. We had this blue thing here, which I described as a nice hug, but it's formally described as an immobilizer. So it's just a little beanbag, really. They lie on it and you can suck out the air, so it becomes rigid. So when the child falls asleep, you just literally pick them up from the pack and play. You put them onto a trolley, which they would look something like this. And then the image over here on the right is showing them with mini muffs on. So they go into the head coil, they have mini muffs on, you've got little pulse socks on their toe, so you just know everything's okay. And it's quite straightforward, especially, I was surprised, even toddlers don't really wake up. If they're 4 or 5 and they've fallen asleep, they're pretty much out. So we had to do a couple of things. So the MRI is a loud environment, so we had this sound cancelling headphone, you put kind of baby socks over the top, so it's quite soft. And then this black roll-up thing is something you have to actually put inside the scanner, so it's more sound dampening. What they've done in St. Thomas's is they have, so the head coil is how they acquire the image, really. And they just made a custom one, sized for a child's head, but that's a very expensive way of going about it. That was for a bespoke study. So as I say, pre-scan preparation is everything. With slightly older kids, so colleagues of mine have been collecting data in 6-year-olds and 8-year-olds where they're doing a task. They tend to use, I guess this is similar to any study, reinforcers. So like if they've tested out being still because it's very motion sensitive, they get stickers and things like that. But one of the things, we had a few incidents, for instance, with kids who really wanted to take part but blatantly didn't want to take part. And you could roughly tell this from non-verbal behaviour, but it's one of those things you have to be quite careful with. And then you show them things like you show them a teddy bear after it's been in the scanner, things like this. You show them a picture and you take a picture of them with the camera moving around and you see they're all blurry. And what we had to do for some children, older kids especially, you put up like some sort of thing around the scanner. So this is like a rocket ship. We had a fairy tale castle. We had to wear either NASA suits or princess dresses, or strangely, I haven't included them. But yeah, so you ended up having to put in a bit more effort. Even at recruitment, you tend to do things like this just so that they'd be interested. But even so over here is a plot of how much they moved, and the actual measure doesn't matter too much, but you see if they're awake, they move far too much, so that most of those scans are pretty much unusable. And so most of the scans I'm going to talk about today, in fact all of them, the children were acquired awake. Asleep. If you look over here on the image, you can see kind of a sagittal slice going through someone's head, and one is with motion correction on and off, and you can see if they are moving, the image slowly but surely becomes unusable. And a lot of these subtle artifacts that come up, that's not very subtle, but subtle artifacts that come up with motion could be masqueraded as a change in development, especially if it's overlapping with age. So yes, you're in an MRI. What are you going to do to measure this development? So one thing that maybe gets lost a little bit is that MRI can really only measure water. So it measures the relaxation properties of water in response to a magnetic field. And those properties change depending on what's surrounding. So if you've myelin, the rate of relaxation in response to a magnetic pulse is faster. If you have free water, it's much, much slower. And what you're trying to do is separate out the rates of relaxation. I guess you've seen MRIs before. This is my head, which is fairly distinguishable purely from my nose. You can see it here and you can see it down here. But the general idea is I will be showing images that mostly look like the green one here, where it's sliced through. So it's axial slicing, because I tend to be more interested in things like the thalamus. Over here is the type of technique we're using. So this is, MRI also doesn't tend to be quantitative. So it tends to be, everything's on a relative scale, but you can acquire images in such a way that they are quantitative. It just takes longer. So the reason we focused on structural imaging, this scan takes about 20 minutes by itself, whereas a normal structural scan is probably about 4 minutes. So what we're doing is we're trying to separate out intra and extracellular water, which we've decided for the purposes of this study that we don't care about. We've separated out free water, which might be CSF, so fluids that just aren't interesting, and we're focusing on a specific population of water trapped between the myelin lipid bilayers. So this is very short T2, and comes up as basically the white contrast on the image over there on the right. So the more white it is, the more likely it is to be myelin. Physicists have a tendency towards making silly acronyms for different things. So like a scan can be called a flash. This one's it was originally called DESPOT, and then they made it multi-component, so it's McDESPOT. And it's fairly standard technique, I guess, but there are lots of variants on these kind of measures. So white matter imaging, I think there's a conference going on as we speak, looking at white matter imaging and methods. They tend to correlate with each other, usually. So have you seen kind of images of, these beautiful kind of spaghetti tubes for tractography? So some of the measures that you get from that are shown here, and actually it doesn't really correlate that strongly. It correlates quite well with things like axonal density. So the degree of myelination correlates with the density of axons, and that makes sense because that's how the brain fasciculates. And it correlates really well with T1. So if we just say it's T1, which is the relaxation time of water in tissue, that's fine. So for validity, so this is an image from Sean Dioni, who's my boss over at Graham. The top row is what the in vivo maps we're getting at specific ages. So these are predicted ages, and the bottom row are from FlexSeq, which is 1909. So as I say, it's depressing in a sense, because really we're just reproducing what we already know. The advantage of having it in vivo, though, is you can fit models to it. So That curve on the bottom right, you're going to see a lot, but I guess you've seen in everything. So that kind of growth curve comes up in bone length, comes up in hair, comes up in how trees grow. In this case, it's a Gumpert's growth model, and it's pretty much every bit of the brain develops in that way. It might be volume, it might be myelin. The thing that differs is this lag time. So if you were in an area of cortex, this curve might start here, and it might go up a little sharper, or it might go up less sharp, or something like that. And most of what we're looking at is how sharp or how quickly things go up. What you can also do, because you've got a model, is you can play it as a video, so you can say, as you move through time, certain regions of the brain are developing at this rate. You can look at independent data sets, and you can show that, first of all, the residuals of this model are normally distributed, so it fits on relatively well. But then you can also, at every point throughout the brain, ask, are these children different? Is there anything different about what they are to what you would expect from a population model? So the actual studies I'll be talking about are really all the three roughly the same study. They're using mostly independent samples, but slightly overlapping. And it's partly because the reason we're kind of reiterating things is we weren't entirely convinced. So as I say, we're using the Mullen scale, so we're looking at individual differences in myelination in these infants or in these toddlers, individual differences in myelination and behavioural scores. We're using the Mullen score, which it's very similar to the Baileys. These are they're more clinical assessments for behavioural development, but they have subscales, so they have things like receptive and expressive language, they have visual reception, and gross and fine motor. Some of these things are floored and some of these things are ceilinged. If you were to look at this with respect to another common measure like the Baileys, which is what they use mostly in the UK, they correlate pretty strongly in language. Visual reception is almost completely uncorrelated, so I don't know what the two measures are looking at, but and fine motor is roughly the same. So I showed you that curve. That curve makes fitting most of the statistics I do standardly really, really difficult. So I'm going to talk about asymmetry of the brain, which is interesting in language and lots of different other areas. But really, the reason I'm using asymmetry is as a fudge. So what we want to be able to do is linearize the problem. So if you look at that curve, if you imagine there's slight individual differences in this curve between left and right, and you subtract them, the nonlinearity is taken out, and you have an asymmetry measure. So what we're looking at is the difference between left and right. So does the rate in left brain development differ to right brain? And in this cohort, we have about 108 children. They're aged between a range of 1 year to about 6 years. The handedness where it's been established is there, but we didn't really find any associations with handedness, so it doesn't really come up again. They're all typically developing, so there's no reason to think that there's any problem with these children. And the reason we're looking at kids a year and above is because below that age, the measures we're getting are quite noisy, and if you do an asymmetry measure between two noisy measures, you just get even more noise, you're just amplifying. So we just ignored the first year. This is relatively standard for asymmetry analyses, so they do this they might be doing it in language, but it's really just left minus right over left plus right. So you have an image in standard space, you literally flip it, and you subtract. And we use non-parametric statistics like this permutation testing because in most cases you can't assume normality in any imaging data. And these are the regions we get. So the orange here is indicating regions of significant asymmetry. The top row is regions of left asymmetry. The bottom row is regions of right asymmetry. So you can see that most of the asymmetric regions we see are leftward. And that's actually quite a lot of these you might maybe expect. What we were hoping to see was something like Wernicke's area, Broca's area, or something like classical language to make life easy, I guess. We didn't really see that. And actually, if you do this measure in older children, you still don't see that. So it might be that that's more reflective of cortical development and not the white matter development we're looking at here. If you look at these regions and you click them out, you see that those growth trajectories we were seeing earlier pop up. So that's normal. You see a couple of outliers down at the bottom, but basically the brain is developing at the rate you've seen from that previous curve. Asymmetry isn't. Asymmetry is pretty much normally distributed throughout the lifespan. So it's a trait in this sense. So it seems to be pretty constant from across individuals. What we're more interested in is to see whether any of these things correlate with what their behavior at the time of scan is. So if you do that, we were doing ANCOVAs in this case. So we're looking at the association between white matter asymmetry and these very coarse measures. And because of the Johnson studies and things like that, we were just as interested in interactions between cognition and age. So does the association between structural brain things and cognitive change as a function of age? So you're going to see these things because there's no actual good reason to assume it's fixed. And we did normal multiple comparisons. So the asymmetry is functionally relevant. Mostly. So depending on what age you are, you have a pretty different association between structural asymmetry, which seems to be constant throughout the age span, and behaviour. And there's a couple of things that would make this a bit sus. So what you'd want to know is whether test-retest in behaviour is good, and actually in these subjects it's pretty good, it's about 0.7. So if you were to acquire the data, and we have this data when they were like 3 months and you have it at 9 months, the standardized measure gives roughly the same score, so it does okay. But what it does kind of emphasize is that quite a lot of the cohort studies, including ones I work on, are looking at brain behavior relationships at a single point. And if you do that at a single point, you get a completely different response to when you do it later. And it does. So if the behaviour is constant and that trace seems to be fixed, it calls into question, to a certain extent, the use of imaging here. Because all that the images have done really is muddy the waters. So if you look at this asymmetry longitudinally, there's no difference over time. So the image is up here on the top. So we've got 69 kids who've been scanned at 2 time points. The asymmetry is the same, but the relationship's changing. So this is slightly misleading on the top in that the thresholds slightly change whether a region is asymmetric at different points, but if you were to look at the raw statistics, it is the same pattern, and certainly there's no significant difference between time point 1 and time point 2. So the behaviour seems to be reliable, and the structure seems to be reliable, but they're showing different relationships at different time points. And that becomes slightly more difficult because, I suppose I'll get back to that slightly later.
Could I just ask you a question? When you say the behaviour is stable, because I mean, there's very little behaviour is stable over that period of time. So what are you talking
The standardised measure through Mullins. So when I say that, I mean there's
so
score.
you're talking about them at a norm. Okay, no, that's fine.
No, I only mean standardised score. But even so, we've. Like some of the things that you'll notice with things like Mullins is, and actually probably every behavioural test, you change psychologists, the scores might shift suddenly, or they might go down suddenly. We were relative, so at any time there was any change, there would be an overlap of about 3 or 4 months between the two till they moved to a point where they were getting consistent scores. But it's a really rough measure of developmental quotient, and actually the range, I'll show you in a second actually. I'll show it to you now, in fact. So when we put this in, one of the reviewers kept asking about ceilings. And this is in the paper now. So they were saying that there was a ceiling on the test, and I've been looking at the standardized scores. So there's no real ceiling on the test, but that's by design. Actually, as you go up in age, so the range might be like, let's say, I'm going to arbitrarily say 30 to 70, 50 being the middle. And as you get older, the range goes down. So you can't have really high developmental quotient when you're 5. whereas you could have had a really high developmental quotient when you're 4. So they pointed this out, and we were able to, that was terrifying. You were able to show that actually if you just kept the earlier age points, it was probably okay. But that's quite worrying. None of us, for instance, not many of us in the department were developmentalists. So we had a clinical psychologist who specialised in children, but older children. So some of the standardised scores we'd be looking at, it took quite a while to get kind of on top of what the variability is, what things like this that happen. So luckily a reviewer pointed this out. So prior to this, by using asymmetry, I'm kind of avoiding the problem. So I'm trying to get around looking at these growth curves. And we're still not talking about growth curves. Growth curves, I guess, formally are when you have multiple points. So pretty much everything I'm doing here is cross-sectional till the very end. So in our case, we decided to go back to the raw data. So we're not going to look at asymmetry anymore. We're actually going to look at those developmental quotients. That curve you saw for age is a log curve. So we put in log age as a covariate later on. We doubled the sample size, and we reduced the amount of kids at this older age where the ceiling was apparent. And we're doing roughly the same thing. The only difference is there's a quirky image analysis step here. So I guess you'd be aware of principal component analysis and things like this. Factor analysis, just to reduce down the data. So another method of doing this in imaging is to use independent component analysis, and in this sense we were just doing it as a pre-processing step. So there's MATLAB packages that do this, but this means instead of doing a test at every point in the brain where there's like 100,000 points or 200,000 points, we reduce it down to a set of independent components that are all just spatial maps, really. But for each of these spatial maps, you get a single score for each subject. So you go down from having 100,000 to about 50 tests. And then your multiple comparison problem is significantly easier. And when you run independent components analysis on this population, you can get patterns like this. So these are, over here, the images in orange are the independent components. And you get bilateral auditory cortex, you get bilateral visual cortex, and actually what all these patterns are actually showing are thalamocortical bundles. So they all myelinate at slightly different rates depending on the process that they're associated with. It splits up the corpus callosum into 3 bits, which you would also expect. If we go on, you get lots of structures. So it is structured. You get artifacts as well, and because they're so blatantly edge-based, it makes it a bit easier. And you also get lateralized parcels. So that's saying that those 2 regions are moving at a different rate, because otherwise, whereas the thalamocortical parcels are moving at the same rate. And this is going to be exactly the same analysis as the previous one. So instead of doing it on asymmetry, we're doing it on each of those independent components. So in this sense, this is probably hugely interesting. The only difference is that because we're looking at these growth trajectories, we're including, instead of a linear form of age, we're including non-linear forms of age, so square and log of age. And we're trying to see whether there's any association with all of the Mullen scales except for gross motor and the Myelination measures we have. And these are all of them, so there are 54 independent components, and we've got 22. So 22 bits of the brain have a pretty obvious association with the cognitive scores, mostly expressive language. And expressive language, if you do factor analysis, is like it's the thing that loads highest on a kind of a developmental quotient. And expressive language in a 3-month-old is a bit messy, but you see the same individual variation, and you see where you see there's a broken line going around, that means there's an age interaction as well. So the association is changing with age. And if you match it up to the previous paper, hopefully, don't do animations, you will see that roughly the same regions are coming out in both analyses. They're independent data sets, bar 20 kids or something, but there's 180 in the second one. So you're more sensitive to weak effects, but you're still seeing the same effect that we saw in the previous study. So that's not that helpful. The only thing that's really helpful, so this is something Sean did as a follow-up, is that the developmental trajectories are different. So this is using a median split of kids according to their developmental quotient, so a combined score of all the Mullen scales. And in the early days, there's not a huge difference. And as you go on later, there's an increase in the gap between those who are doing well and those who are doing poorly. And actually, you could imagine this to be that moving average correlation as well. So basically, as you move up, and the kids get older, there's bigger range. And only by having the bigger range, you're able to actually fit those correlations in the first place. So the reason we're seeing that moving average and that change is partly due to the range of the data. It's not entirely, so you can still separate them out. But it's beginning to say that from what we can measure, some of the anatomical bits we're seeing aren't that informative at an early age as to how they'll do later on. So asking where this goes is a bit rough. So the reason we were doing this in the first place was to provide a baseline. So the end point isn't to scan 2,000 typically developing kids. It's not particularly useful. The end point is to start trying to look at how kids who deviate from normal development and see whether there's anything that's predictive of their outcome, if there's anything that would be predictive of whether they respond to treatment, whether they respond to medication, whether they respond to anything. And the one thing that came out of this was a normal study, so we have normal parents, there are lots of variations, so they're from different socioeconomic status, different home environment, everything. So you're able to start looking at things that are a bit more speculative. So this is probably the most controversial of the topics we were doing, which was just doing a really simplified analysis of children who were breastfed, children who weren't, and children who were both. And you can find differences in myelin, and there's a reason that would be interesting, because quite a lot of the fatty acids that are associated with breast milk are associated with myelination, but these are tiny differences. So just like the behavioural differences we were seeing earlier, the range is quite small, so we're looking at really small differences depending on outcome. But actually it's been replicated a few times, and it seems to be, there's quite a few studies specifically looking at early child nutrition and brain outcome, not necessarily behaviour, whereas behaviour might be more powered. The one that's a lot fuzzier is individual differences and socioeconomic status. So the image up there on the right, that's covered by brain, I think, is just showing that there are different trajectories of grey matter in that case, and how they develop depending on the child's home environment. This isn't, I don't think it's controversial. The image below is some pilot data we have showing the same thing. But from the group I'm working in, so we have children who were born preterm, and we followed them up, and they're from a very distributed background. So Gareth Ball was working on this, and we have a very coarse measure of socioeconomic status, and that is their postcode. So UK is very detailed postcode. So and in those children, parental socioeconomic status, as it said here, was the single largest predictor of outcome. In children who are born preterm. So it's nothing to do with the clinical factors. So these are kids who were born preterm, didn't have a major injury. And that's quite severe. It's good in a sense in that it's one of the things that can be changed, but obviously not particularly easily. So I guess that's the last slide. Single MRI is only a snapshot, so when you scan and when you look at a child, completely overwhelms what you're going to see. So that it's almost definitive. So longitudinal data is probably the way of doing it. It's very difficult to do longitudinal data. So if you look at some of the sites especially in cities, the parents might be there when they're an infant, but following up when they're 4 or 5 is quite difficult. In Providence, this wasn't so much of an issue, as people tend to stay in Providence once they move there. They actually tend to move there for families, I guess. The other thing is, so the effect sizes I'm talking about when you're seeing correlations are tiny. So they're variance accounted for of like 4 or 5%, and cumulatively it might be 9 or 10%. some of the bigger studies, so there's a UK Biobank study where they're looking at about 10,000 people, and their early stuff is coming out, and the percent variance accounted for when you start taking into account SES, nutrition, health factors, it goes down to 0.5 or small, so the kind of effect sizes you'd expect from genetic studies. So a lot of the imaging studies that have been done prior are tremendously underpowered for the effect sizes they're showing. And like I was doing a moving average correlation just to show. So, but it's kind of remarkable how little effect some of these things, some of the brain effect or brain measures have on outcome. And that is it, I think. So these are all my funders. It's lots of pictures, and that's the logo we have to put on we had to put on our business card. So it's nice. Okay.
Johnny, we have time for a few questions if you're
Sure.
Okay. You want to ask Johnny a question?
I know you chose the Mullins because obviously it's normed across the whole age range, but when, as the study's ongoing, do you think that you might add in tasks from children aged 3+ in terms of looking at frontal lobe functioning
So-
in particular?
They have, so I've that group is now split. So the group that was in Brown is now in Denver and in Brown, which is just to make it cross-continental. But they have started adding stuff. So for instance, you start moving into Wechsler age ranges when you're at like 4. What they've also started doing is, like you're saying, frontal tasks. So like response inhibition. But even the response inhibition tasks between a 4-year-old and a 6-year-old, there's 2 different normed versions. And they seem to be quite explicitly different. The interest kids have. Like, we were trying with follow-up on the preterm children, and there's a floor effect for quite a lot of the preterm children compared to healthy children and things like this. So it's really, really difficult to get measures. We have started moving into things like working memory tasks and things like that, but they aren't necessarily, I don't think they have enough subjects yet to be able to start doing much with them. But it's remarkable how little consistency you necessarily get across the age span with some of these tasks. Like Bailey's, I think, goes up to 4 years, and Mullins is supposed to be up to 6.
Yeah. So I guess they're developed for clinical purposes. So,
Yeah, exactly, yeah.
you know, but with the response inhibition task, is that computerized measure or is it
It's computerized, or yeah, the one they were using when I left anyway is computerized. So most of these kids can do touchscreen tasks quite well, so they've started moving towards touchscreen based ones, and they're really good at it, and it's a much better measure of whether they can do it or not. Like even with the Bailey's and Mullins have an actual telephone. It's one of the pictures they have to recognise. None of the kids would have seen a telephone with like a spin thing, so they're a little out of date.
John, I was interested in just thinking about the preterm and attachment, I suppose, and the impact of attachment on, I suppose, neurons and the synapses early on, and the showing now. And I just thought about that, about classification of attachment.
So they don't. So I guess quite a lot of the Bill and Melinda Gates Foundation, for instance, are very interested in that, or they were at a conference certainly, but the measurement of attachment takes quite a while, and they're trying to push towards sample sizes of like 10,000 kids, and they've had trouble with that. What they've been focusing more of in the preterm group is things like the actual environment. So the children will have lots of injections and things like this. There'll be lots of noise around, so the environment itself is very different, and that's something they can measure. Whereas the attachment in our group, because the group that I'm working with at the moment anyway is neonatologists mostly, so they do follow up, but their endpoint is what happens to the preterm child. It's not necessarily what happens to the child later on. That moves to groups we're collaborating with.
Johnny, question about, the effect size problem you mentioned. Does that make you question any findings that are reported in early neuroscience studies?
So this is another of those controversial things. So some people are saying that the early reported findings, well, yeah, actually, I guess everyone does, but the early reported findings were big effect sizes, so they're likely to be reproducible. So if you think of something like someone in a scanner doing a task, I think they're probably reliable enough. It's when you start comparing groups. So if you're comparing a group with and without some behavioural variable. If you're comparing disease groups, it's probably okay. So if you compare the effect size for the size of the hippocampus in someone who has temporal lobe epilepsy, that's fairly solid. You'd probably see that in 20 subjects versus 20 subjects. The effect size in the difference between 20 subjects who have an IQ of 110 versus an IQ of 90 is probably going to be tiny. So yeah, the association in adults with IQ and brain structure is probably the most controversial area. Largely because there's a lot of, I guess I remember this from undergraduate, but a lot of controversy over the measures themselves, and even how they do factor analysis on them, because depending on how you do factor analysis, you get completely different brain correlates to the intelligence. Imaging has been taking a lot of flack recently, and I think quite a lot of that is justified. And I think, in fairness, they seem to be coming to a consensus of just doing things a little better. So making sure data is replicated, making sure things that you would normally have in a grant. So things like make sure you replicate someone else's effect before you move on to a new question. And I think that's getting better. I'll be optimistic anyway. Every 2 years or so, there's a new paper that says that it's all wrong. Everything that's been done previously has been wrong. And everyone gets in papers, go around for a little bit, and 2 years later, when everyone's kind of forgotten, there's a new paper that says it's all wrong in a different way. There was a paper recently about how there were very high false positives in imaging analysis, and that's entirely true if you do it in a certain way, but that was also published in 1997. So it was done a long time ago. It's just that it tends to be forgotten and then remembered, and forgotten and then remembered. But that's the major reason towards this move towards big samples. I don't think there are any studies in the IOP, certainly at the moment, which are under 100 subjects. And that's even with disease, where you would expect big effect sizes. So hopefully it's getting better. It's very expensive to do on large samples.
Can I just, in terms of the language skills and so on, were these kids all monolingual in this group, or were some?
Oh God, no.
So how is that taken?
So if there's so Providence is quite multi-ethnic city. It's a university city as well, so you have people from all sorts of different backgrounds. Usually, so with quite a lot of the early infant stuff, it's more of an issue. But some of them are very basic sensory questions. If there's any actual language, they might ask the mother to do the question, or they might have someone who speaks the language in with them. And that tends to be the way. Once they're toddlers, it's probably okay because they're used to television and things like that, and their knowledge, it's not as language dependent as you might think. Quite a lot of things are just you throw out toys in front of them and see what they do with them. So part of it is observational. But it is messy. We did look on language background, and it doesn't have an obvious effect on the imaging data or the imaging associations with behaviour, which again makes you wonder what they are because it should have something. So there was Michelle, who's not on here, but she was one of the undergraduates in her group who was looking at bilingualism versus monolinguals, and there were effects, but they're very subtle. They've been in the ventral temporal lobe, and I don't think they quite reached statistical significance, so I don't think they were enough to mean anything. So I think it's safe enough.
Maybe one last question, if anybody has a comment or question.
The association with breastfeeding, I found really interesting. Was that just a raw difference, or did you control for anything?
No, in that study,
socioeconomic factor.
Yeah, so they were matched on socioeconomic status. They were matched on about 5 different things because at the same time as the breastfeeding study was being done, they were also doing a study on socioeconomic status, and they realised there were differences in both. So they were trying to match across the thing, so it turns into a very elaborate ANOVA. So they were matched on basic demographics. There's always more to it, especially with things like nutrition. So that's why I think it was put out as there were probably about 200 children in the study, and it was mostly longitudinal, but it was still described as a pilot study, and I think it still was a pilot study. Because it does have quite a big effect. I think there are follow-up studies currently going on.
And do you distinguish between exclusive and non-exclusive?
Yes. So there was 3 different groups. There was exclusive. I think they stopped a group that stopped at 6 months or maybe 3 months, and a group that exclusively bottle fed. But like there are other, especially with exclusive bottle feeding, there are other factors that lead to that. Some of them clinical, like they're just the child didn't take to it or something like that. So there are. It's more of a. That's why I kind of left them as short topics.
Well, if they had a C-section, that would predict that had a big effect.
I think they were. So I can't tell you that for sure because I don't remember. But I think they were matched on the mode of delivery. Well, C-section doesn't preclude you from breastfeeding.
No, but it has an effect.
Yeah.
Quite significant.
Does it?
Oh yeah.
I'm fairly sure they're matched on mode of delivery.
In what way? Does it?
What?
In what way?
So if the child is delivered by C-section, the child is much less likely to be breastfed.
But so I saw preterm infants less likely to be breastfed than typical full-term birth, because they're weaker and smaller.
Yeah.
They might be breastfed from expressed milk, but then they're very unlikely to be feeding naturally in the very early days.
That I actually don't know.
Yeah,
And the
I
desire
think they're too small, they're too weak to
Yeah.
latch on.
Well, I guess when they're in the NICU, they can, but.
But I know that they do encourage mothers to express, and then it's fed to them, but as bottle, and then it's very hard to transfer those babies to breastfeeding later.
Yeah. Yes, I would imagine. Yeah, no, I didn't know that actually.
I think there's such a push for them to put on weight quickly that medically they want to substitute breast milk all the time with stuff that will put fat on quick. In preterm babies as well. So it does.
But it would introduce a potential confound in that the preterm babies breastfeeding are likely to be the stronger of the group, or with the higher.
Well, so that study they were in preterm. So these were all they were all typically born. So the preterm work is the stuff done at King's now. So this is all the stuff at Brown. So kids who were preterm were excluded from all of these studies, I think. But there was a different cohort of children who were born preterm. But that almost certainly would have an effect later on.
Okay, I think the room's being used at 2 o'clock.